Hello, this is a brief sketch of the reverse transcription polymerase chain reaction, or RT-PCR for short. This method can be effectively used to identify the presence of active viral COVID-19 RNA in a patient. So let's get to it. First, viral RNA needs to be gathered. This is typically done with nasal swabs or even spit samples. Although the spit samples are less effective due to a lower viral load, this is less intrusive and quick for surveying across a large population of people. The sampling pool can include many people, but more RNA is needed to detect a viral presence, so scientists use reverse transcription to convert RNA into complementary DNA. The sample from the patient is added to a test capsule along with loose nucleotides, complementary primers, and the enzyme reverse transcriptase. The complementary primers only attach to the N gene segment of the virus's RNA. The N gene is responsible for the nucleocapsid protein, a structural protein in the virus that bundles the RNA. Once attached, these primers act as a signal for the enzyme reverse transcriptase to fill in the rest of the strand, almost like a zipper that is constructing itself. This new strand is the complementary DNA to the RNA. The complementary DNA still needs to be replicated, so we use the polymerase chain reaction, similar to reverse transcription, but we are converting DNA into more DNA. This is called amplification. Additional specific primers are added to the complementary DNA sample, along with the appropriate nucleotides and the enzyme Taq polymerase. There are 3 steps to the polymerase chain reaction. The first step is denaturation of the complementary DNA into 2 separate strands. This is done by raising the temperature, which interferes with the hydrogen bonding between the strand base pairs. The second step is annealing. Specific primers are added at a lower temperature to ready the complementary DNA strands for the next step. The final step is extension. The Taq polymerase enzyme is activated by the primers and replicates perfect copies of the complementary DNA of the viral N gene. This cycle is repeated about 40 times to acquire a sufficient amount of viral complementary DNA. 40 cycles may sound like a lot, but the entire process can have a realistic 24-hour turnaround time from collecting the sample to getting results back to those who were tested. Finally, complementary fluorescent probes are added to the complementary DNA batch. These probes are unique and only emit fluorescent light if bonded to the viral N gene. The amount of fluorescence determines the concentration of viral RNA in the sample. A high level of fluorescence indicates a positive test for COVID-19. Note that this test only indicates if the samples are positive with active COVID-19. It does not indicate whether or not the tested have had COVID-19. That would be an antibody test. Well, thank you for watching, and remember, stay safe out there.